we've had some really heated conversations in the Genetic Evaluation Board at LactaNet about inbreeding. And in a couple weeks, I'm actually going to the American Dairy Science Association Discovery Conference in Naperville, and we're going to talk even more about inbreeding there. It's a hot topic, and it's a pretty prickly one because nobody really knows how to solve it. So what I think is really good is that we can have some discussion. I can give you some background information on what it is, what it does, and what we can do about it maybe. So the outline of my presentation today, I'll give you some basics on genomics. I know everybody loves when you get taught stuff, but I hope that's okay, just to make sure that we're all on the same page. And then we're going to talk about what is homozygosity. So just a question, how many people know what homozygosity is? All right, how many people know what inbreeding is? Everybody knows what inbreeding is and nobody knows what bloody hell. All right, so it's the same thing basically, okay? It's just a fancy word that we try to use to make sure that people don't think that we're inbreeding our cows. That's basically what it is. So we're going to talk about that though yet. So I'm going to talk as well about some recent studies that we've done in my group at the University of Guelph on Canadian dairy cattle, looking at things like the rate of inbreeding, how is it changing, the effect of inbreeding on production and fertility traits, and localization of homozygosity. Now those are really big words. What that basically just means is in the DNA, where are there spots that are homozygous? Where are there spots that have no diversity anymore? And then finally, a question that I'm actually going to ask you guys is what do we do about this? So first off, some basics. You might have seen this before. Anybody seen this before? Yeah? All right. So you know that the phenotype, right, which is sort of something that we can measure or see on an animal, right, something or anything that you can measure or observe, is equal to the genotype plus the environment. So there's two sort of main things that affect how an animal, what an animal can produce. And we've got the genotype, that's the animal's genetic information, and an environment. And that's how the animal is fed and housed and managed, etc. And geneticists, we always get a bad rap, right? Because geneticists in the past were real jerks, and all they wanted to do was say genetics is everything, right? And as we all know, that's not quite true. If you think about what heritability is, heritability, when you have a trait, is what's a good heritability? Does anybody know?
25.
Stature. Brilliant. What's the heritability of stature? High. 0.4. Does anybody know what the heritability of human height is, actually? Because we can have heritability with humans as well. Eric, you're pretty tall. How tall are your kids?
They're small.
Are they really? But okay, they're young, Eric. All right, who's the tallest guy in here? How tall are your kids?
Actually, taller than I am.
My son's 6'8", my daughter's 6 foot.
How tall is your wife?
5'11".
How tall are you?
6'3".
More than average. Taller than average, all right? So heritability of height is pretty high. The heritability of human height is like 0.8, I think. And for cattle, stature is really a highly heritable trait. So if you're tall, your partner is tall, your kids are probably going to be more tall than average. And the same thing is true for a lot of other traits. But heritability for some traits is lower, like milk production, 0.4, 0.3, somewhere in between there, and it changes according to the population that you're measuring. So sometimes genotype has a huge role, and sometimes the environment has a huge role. And sometimes there's even interactions between those two, but that's too complicated. We don't want to talk about that. So today, we can measure the phenotype really exactly, right? There was a whole bunch of conversations this morning and talk about SNF, right? And protein and fat content and lactose and all that lovely stuff. We can measure a heck of a lot more than we used to be able to measure, and a hell of a lot more accurately as well. With regards to the genotype, now we can actually look at the animal's DNA. And with regards to the environment, we've got quite a few changes that have occurred in the past 50 years with regards to housing and management and feed, right? So the genome is composed of 4 nucleotide bases, adenine, thymine, guanine, and cytosine. And you guys have all seen this wonderful DNA helix stuff, right? You know what that is. But what's really fascinating is that all life forms on this planet have a blueprint written only in these 4 letters, A, C, T, and G. Okay? And a gene, when we talk about genes, it's just a very specific sequence of bases of these letters here. And some genes act as instructions and some don't. So when we talk about DNA, it's actually a lot more simple than some people will have you make out to think, right? It's just a letter, 4 different letters in a certain order. Now, we can see here that DNA always happens in pairs. And you get 1 strand of DNA from your mother and 1 strand from your father. Okay? That much should be clear to you guys already. Are you with me? Who did I lose already? People who get lost get shot with the laser, just so you know. Right between the eyes. I'm pretty good at this. I've got like lots of experience. So the basics of genomics, right? We've got, anybody hear about the coronavirus? What's that? Yeah, exactly. The coronavirus is an RNA virus. It's got about 29,000 base pairs. That means that if you took its DNA, took like a meat flattener, and you hammered it out, it would be 29,000 ACs, Ts, and Gs long. That's it. Now, E. coli, heard of it maybe? Have it maybe? I don't know. 4.6 million base pairs. A chicken, about 1 billion. What about a mouse? 2.5. What about a cow? 2.6. What about a human? I can't draw very well, but this is supposed to be me. It's maybe no surprise to some of you that I have about the same amount of DNA as a cow. Some might be surprised about that, some might not. But in case anybody is wondering, humans have about a little bit more DNA than cows do. And that's pretty surprising for some people. For some people, it explains a lot. So now you guys know all about genomics, right? So let's look at some cow genomics. We've got 2 cows here, Sally and Susie. Sally and Susie both have about 2.6 billion base pairs, 2.6 billion of these letters, right? Does anybody know how many base pairs do Sally and Susie have in common? Lots? Who says they share more than they differ? Who says, wow, they're probably really different? Yeah? Sorry, you're wrong. They're pretty much 99.7% identical. All right? That means that there's, believe it or not, still 7 million bases between Sally and Susie that are different. And those differences, those individual letters that are different, that is what we call SNPs, single nucleotide polymorphisms. That's a massive amount of work, right? Who's ever heard about a SNP? Did you know that it was called a single nucleotide polymorphism? Yeah? Perfect. Wonderful. Who didn't know that? Nobody wants to admit that. All right, so that's fine. So I'm telling you now, let's call them SNPs, okay? Those are single nucleotide polymorphisms. Believe it or not, that makes sense because they're single, they're individual letters, they're nucleotides, that's what they are. And polymorphisms just mean that they're different. Okay? So these 2 cows have a lot in common. Now, at 90 days of milk, Sally makes 90 kilos of milk and at 3.5 protein and 6% fat. And Susie makes 3.5% protein and 5% fat and makes 90 kilos of milk. Which cow do you keep? You've only got 1 spot. All right, you've got 1 spot left over. Which one do you keep? Guys, this is a no-brainer. Who's pregnant? Okay, they're both pregnant. They both eat the same, they look the same. Sally. Sally, right? Clear. She's got 1% more fat. Perfect. Beautiful. So you're keeping Sally, but the reason that Sally might have 1% more fat is because of her DNA. When you go back to those single nucleotide polymorphisms and you look at the difference between Sally and Susie, and you match up this difference in phenotype with the differences in the DNA, you can start to identify where they're different, which is pretty cool, actually, because that means that you can choose, you can look at this DNA the day these 2 animals are born. That means you don't have to raise, even if Jersey heifers live apparently on air and get fat anyway, kind of like me, they really do, it's still a spot in your barn, right? So if you can make that decision earlier, that's pretty cool. So you don't take Susie. My sister's name is Susie. She'd be terribly sad about this, but you're not going to keep Susie. So why is this genomic information important, and how has it helped us? Well, the gain before genomics, you can see here on this graph, right? I've got a whole bunch of traits written on the x-axis here, and on the y-axis, one of my grad students did some fancy math to make sure that you could compare all of these different traits on the same scale. So normally you couldn't compare milk yield and somatic cell score on the same scale, but we've done it now so that we could compare those two different traits. We had some gain before genomics, but there were some traits that were pretty hard to change with genetics. But after genomics, in the last 5 years, we've seen across the board a really good increase in genetic gain, which is fantastic, right? Awesome, great. Everybody's happy, making lots of money, everything's fantastic. In particular, the most fantastic thing about genomics is for traits with low heritability. Remember that term that I used earlier? We were talking about heritability, how much genetics is actually behind what we see. Well, here you can see, again, on the y-axis, standard deviation units, so they're comparable. On the x-axis here, we've got year. And here we've got two traits, herd life and somatic cell score. Herd life, between 1994 and about 2002, herd life was decreasing. And somatic cell score was increasing. Is that good or bad? Bad. Okay, so decisive moment in dairy cattle breeding history here. Herd life and somatic cell score EBVs were included in LPI in 2002. Now, the trend for somatic cell score started to decrease, and the trend for herd life started to increase. Fantastic. But what happened in 2009? It says right on the slide, literally, guys, come on. Genomic selection. Genomic selection came, and we started to select animals based on genomics, based on their genomic DNA values. And you can see herd life increased and somatic cell score decreased. Wonderful, fantastic. Why is this so important? And I'm a geneticist, so I have to go back to a formula here again. Genetic progress per year. Has anybody seen this formula before? Does anybody know what the answer is? You're going to hate me by the time I'm done, because there's like so many numbers and formulas and stuff. I love it. Okay, genetic progress per year is equal to the accuracy. How well can you estimate what that true breeding value is? Accuracy, how accurate you are. Times selection intensity. You guys know that, how choosy you are, right? And finally, genetic variation, because you need to have that heritability component in there as well. If a trait's not genetic, you can't improve it. All of that divided by the generation interval. Now, in terms of genomics, we've really changed the accuracy of selection and the generation interval. Those are the two main things that we've changed with genomic selection. So, quick sub-summary here. Animal performance is based on mainly genes and environment. Genomic selection is based on the differences in the DNA, and you can predict that really early. Genomic selection supports faster rates of genetic gain in many economically important traits, and the main benefits are an improved accuracy of selection and decreased generation interval. All right, so that's kind of where we're at today. Now, why on earth am I telling you all of this when I was supposed to talk about inbreeding? Because what we've seen is through these different selection patterns in the past, we've seen changes in the amount of inbreeding in our populations. And you guys know that, right? Who here is even worried about inbreeding? Who's not? Who says, come on, not a problem? Anybody? Yeah? Okay. East Gen. Not a problem. Okay, cool. Then we've got some discussion points. All right, let's see. Here we've got year of birth, 1970 to 2020. And we've got different eras of selection. Okay, you can barely see it, but for you guys, important would be this brown line. This is the inbreeding trend, the average inbreeding level of Jerseys since 1970. All right, so in 1990, right here, BLUP selection started. That's just a method that we use to select animals. In 2003, Lactanet, or at that time CDN, implemented an R value for constraining inbreeding. Whoever looks at R values, do any of you guys look at R values? 1, 2, 3, kind of half. All right, so in any case, you can see what happened when people, when it was new and exciting and everybody wanted to look at R values. It was cool back then. The inbreeding trend it actually helped to maintain a certain level of inbreeding. Now remember, this is average inbreeding level, and you can see how it's increasing. Okay? Now, since genomic selection, we can see this rapid increase again, in particular in Holstein animals. So, genomic selection is fantastic because we can get this increase in genetic gain through reduced generation interval, increased accuracy, and we can select for traits that are recorded in very few animals. Wonderful, I can quit my job and go and, I don't know, do something else. Barbados. Yeah, cool. All right, but, there's a little but here, guys. Homozygosity is increasing at an increased rate. Homozygosity, just imagine it to be inbreeding. Okay, that means that 2 of those chromosomes that you get, one from your father and one from your mother, that they're the same at a certain spot. That's homozygosity. Okay? What is so bad about this? Well, first of all, homozygous recessives, lots for Holsteins, few for Jersey. Has anybody heard of a homozygous recessive? Anybody hear about something like curly calf syndrome, maybe in Ayrshires? Anybody seen a curly calf? Yes. Sounds cute, right? Not cute at all. Not cute, not good. Most recently here, already this year, 2022, the USDA, Paul Van Raden's group, published a new paper on inheritance of a mutation causing neuropathy with splayed forelimbs in Jersey cattle. So that's a new lethal recessive in Jersey cattle that comes up when you get 2 genes that originate from the same ancestor and they come back together in the pedigree at the same time. You have both copies at a certain spot, you're going to have a problem. Okay? Homozygous recessives with delayed impact, those are really nasty. Anybody hear about the cholesterol deficiency haplotype in Holsteins? Terrible story. Such a pain because those calves are born alive. After 3 weeks to 6 months, they all die, no matter what you do with them, because they don't have enough cholesterol in their blood. And you can't see that.
What's it called?
HCD, haplotype for cholesterol deficiency. It's terrible, right? Because you think, oh yes, I've got a calf. And then it just, no matter what the vet says, no matter what anybody does, no matter what you treat it with, it's going to die. That's genetic and you don't know about it. Okay? More subtle effects of inbreeding are also bad. Reduction in the phenotypic mean value and reduction in the rate of selection response. So we're really eroding sort of the basis of our genetic variability that we have to select from. Potential future implications, to be honest with you, I don't know. Financial economic losses, to be honest with you, I don't know. But John Cole and his friends, they figured out that it's about US $11 million per year. Okay, so yeah, it's something, right? I don't want to say that selection can't outweigh that. And right now, it seems that what we're doing, looking for genetic gain, still outweighs the bad side of inbreeding. And until that changes, the train is moving in one direction. All right, homozygosity. Now we're going to get really into the hard stuff, guys. So hang on to your seats, all right? Bear with me. What exactly is inbreeding? Does anybody know? You could say it's the mating of related individuals, right? That's the simplest term. But actually, there's a guy, Sewell Wright, in 1922, believe it or not, he already was thinking about genes and DNA and all this stuff. And he said that he came up with this term called the coefficient of inbreeding, and that's the probability that if I were to take one of your hairs flatten out your DNA and look at some random spot in there. The probability that the letter on your mother's DNA strand and your father's DNA strand is identical by descent. That is your inbreeding coefficient. So it's just a probability, right? The probability that two base pairs at a randomly chosen position in the genome are identical by descent. Now, what do I mean by identical by descent? I mean that they came from exactly the same ancestor. So if you look at this graph here, I know it looks terribly complicated, but just concentrate on this green bit of DNA here, okay? Just so you know, squares are for boys, circles are for girls. So you're looking at this little green strip right here, right? In the offspring, one male son here got a little bit of this DNA. And then this male son, he went out and had two girls. Imagine that, two different girls. And with this girl, he had two kids. And with this girl, he had one kid. And for some strange reason, this daughter and this son, half siblings, got together and did the nasty. Right here. Okay? This is what happens, and this happens all the time in dairy cattle breeding. All right? I have to say some provocative things just to keep you guys awake, okay? I'm Catholic actually, so realized inbreeding right here. You can see that this piece of green DNA came all the way down on both sides of this pedigree and ended up, boom, in one individual. And that is homozygosity. Okay? And that's also inbreeding. So when we talk about pedigree versus genomic inbreeding, there is a difference there. Okay? Pedigree inbreeding assumes that all individuals in a base population are unrelated. We know that's actually not true. It doesn't account for something called Mendelian sampling. You guys don't have to worry about what that is, but ultimately here, the inbreeding estimates are expectations. We expect a brother and a sister to share 50% of their genes. Would you guys agree with that? 50%, right? So genomic inbreeding, if we're looking at actual DNA, we see that they don't always share exactly 50% of their genes, surprisingly enough. Anybody went to agricultural school and took some genetics classes, you know that there's a pretty easy little tricky formula that you can use to figure out what the inbreeding coefficient is. You draw out all the common ancestors. You count how many common ancestors an individual has on both sides. Here in this case, there's 5. And the trick is you say a half to the exponent 5 is 1 over 32. Zulu's inbreeding coefficient here is 3.1%. What does that mean? It means that if I took a gene out of Zulu, it's 3.1% probable that that specific gene has a common ancestor, came from the same origin. Complicated, right? It's not very funny. But watch this. This is even cooler. You've got full sibs, so brother, sister. They're supposed to share 50% of their DNA. And a half sib, which is pretty much the pedigree structure that we have in dairy cattle, they're supposed to share 25% of their DNA. Okay? That means that this animal here is 12.5% inbred if these two mate. So how does that look if you look at the DNA here, right? This animal has 2 half sibs, and they come together and have a progeny. So here we have an expected inbreeding rate of 12.5%. If we have genomic information, though, we could see that actually that shared piece of DNA that comes down through this pedigree could be as high as 25%. It could also be as low as 0%, just by chance. It's pretty improbable, but it's possible. So really, these half-sibs are not all 12.5% related. Most of them are, but you can vary all the way from 25% to 0%. And that information you can only get from looking at the DNA exclusively. So runs of homozygosity, now that we all know what homozygosity is, we can actually measure in the DNA these stretches of DNA that come down and that are identical. Runs of homozygosity, remember this green example here? They're unbroken homozygous chromosomal regions that are present on chromosomes of a specific animal. And they came down into a pedigree, right? So when we start looking at a whole bunch of different animals, we can see a lot more variation in inbreeding coefficient by using this DNA information. Right? And you can see that, maybe you can see that, maybe you can't see that in this graph, but here when we look at pedigree inbreeding coefficients, the distribution is very tight. We don't see a lot of difference because we're expecting all siblings to have 50% shared DNA and all half sibs to share 25% DNA, and we can't really see what exactly is going on. But if we use DNA, you can see here in this FROH, inbreeding coefficients based on runs of homozygosity, we can much better differentiate between animals that are actually inbred and those that by chance don't actually share that much DNA with each other, even though they're related. So why is this all important? You're probably asking, right? You're like, why the hell is this over yet? Come on. Well, on this graph, you can see down here the population size. So you might have a large population over here and a small population over here with a high inbreeding coefficient. This large population has a low inbreeding coefficient because there's lots of diversity there. And then you look at this y-axis and you see a decrease in fitness. And you can see that the smaller the population, it could be, we don't know for sure, but the smaller the population, it could be that there's a huge decrease in fitness at some point if this relationship isn't linear. Okay, if this relationship here is linear, then maybe we can manage everything, but we don't know. Anybody have chickens or turkeys at home? Do you have like any worries about biosecurity there? Yes, right? They're pretty tightly cooped up. You'd have to sometimes even shower before you go into the barn. Does anybody know why we don't have to shower when we go to milk the cows? Because the cows aren't that inbred, right? They still have a lot of variation in their DNA, and they're pretty robust. They can deal with environmental perturbations, but you got to be pretty careful with pigs, and you got to be pretty careful with poultry these days. Just saying, right? We don't know where this curve is because the generation interval for cattle is so long. But for pigs and poultry, we can turn around. We have a much quicker turnaround. So we can make a lot more genetic gain in a short amount of time, but we can also decrease the amount of variation in there pretty quickly. So we've done some recent studies here. Here's just a few of them on both poultry and dairy. And I'm going to go into a few of these studies. Are there any questions, first off? I've probably beaten you up. Yeah.
So, and then you had a graph before that was showing genetic gain after genomics.
Yep.
And you made the comment that we've got genetic gain and we've got also an increase in inbreeding.
Yep.
So do you mean that we can't exactly figure out when we're going to hit
Yes. So right now, and I'm going to say a very nasty sentence. Today, the monetary genetic gain that we can achieve from intense directional selection, okay, the money we make from selecting cows based just on genetics and just on genetic gain, the money that we can make from that today still far outweighs the cost of inbreeding depression. As a controversy-loving person, I would have loved to be able to tell you something else, but it's not the case, right? And we might all know that because you guys look at your cows and you've seen inbreeding rates sort of increase over time, right? And you're like, oh, yeah, but it still works. I'm still making money, right? Everybody's worried about like inbreeding, but I'm still making money. The milk check still comes in. But the problem is that we don't know when this inflection point here is. Right? When does 1% more increase in inbreeding cause a bigger decrease in fitness? And we don't know when that is going to happen.
Is it the type of thing then that we'll know it when we find it, and then we've got to figure out how to handle it?
Well, that's the question. Do we want to go that path? Right? And we've got a lot of extra things that are happening right now, right? Reproductive technologies, AI, right? I mean, I just told you, inbreeding, the inbreeding coefficient was defined in 1922. Maybe we need a new term for inbreeding, right? That's kind of why we want to talk about homozygosity and not about inbreeding, because inbreeding has got so many bad connotations. We wouldn't be where we are today in terms of selection if we didn't have inbreeding. No selection program works without inbreeding. That's the truth. But we don't know where this is going to happen, even if, but I tell you, if it happens, we're in a pile of, thank you.
And how much does it increase if it's generation after generation? It's not just a subtle increase, it increases that much more, correct?
It could, but that's why when we look at these studies, we're going to get to that one, okay? So the first study, I'm going to go through 3 studies, hopefully relatively quickly. The first one is we wanted to estimate the size, the rate of inbreeding, and the effective population size using pedigree and genomic information for animals born between 1990 and 2018. We wanted to look at the changes in effective population size following genomic selection, and we wanted to look at the genetic diversity pre and post genomics. Okay, so what is effective population size? How many jerseys are there right now in Canada? Anybody know? How many? 110,000. Question mark. Cows. Cows. That's a question mark.
I don't know, we have 12,000 registrations a year, say, and they live for, say, 5 years, so maybe less.
Okay. So we had some data available through LactaNet, and we looked at Holsteins and at jerseys, and we had a data set for jerseys of 304,000 jerseys. For jerseys, that's a huge data set. I love it when we can have a data set like that. For Holsteins, of course, we had 676,000 animals, but these are only animals with genotypic information. Okay, so we only looked at the ones that have genotypic information. And we went back for Holsteins 34 generations and for jerseys 26 generations. And for jerseys, we had about 22,000 sires, 214,000 dams, and about a pretty good pedigree completion index. We had 285,000 animals with known parents. So this is a subset of the Jersey population in Canada. Just the only ones, the only animals that we included here are ones that are genotyped. So when we look at generation interval, right? Remember that formula that I showed you with genetic gain? And I said there were those different things that fed into genetic gain. You can tell, here we have generation interval on the y-axis and year of birth of offspring on the x-axis. And you can imagine what happened right about here. Anybody know? Genomics. Genomics has totally changed everything, okay? The time it takes now for the sire of a Jersey bull to have an offspring that replaces itself is like less than 2.5 years. That's how quick that happens. Less than 2.5 years. It used to be like 7 or 8 years. So the train that we're on is going very fast, okay? Now these are Holsteins, right? Do you think this is going to look different for jerseys?
It'll be a little more spread out, I think.
Who says it's going to be more spread out?
It'll be a little more spread
I learned something from Gene. He was very provocative, so I'm going to try and be provocative too. This is what it looks like for jerseys. Not more spread out. Generation interval is even faster. Okay? But don't worry, it's not all bad about Jerseys. I'm going to tell you some good things about Jerseys too, compared to Holsteins, because I guess that's it's pretty cool to do that here, right?
Why is it faster?
Why is it faster? Probably because the effective population size is smaller. So that's the theoretical number of males and females that it would take to express the amount of genetic diversity that you've got in the population. So we've got less animals here, and remember, this is a subset, right? This is not all the Jerseys. These are just the ones that are genotyped. So a very select group of animals, but it's a pretty clear pattern, right? Genomics happen and everything goes faster. That's the takeaway here. All right, so rate of inbreeding. Holstein data each little bunch here is a 10-year period. I've got this fantastic PhD student. He loves to measure everything. So he measured inbreeding like 17,000 different ways. And then I said, you're only allowed to use 6. Okay? So this is what he came up with. So he used 6. Every color is a different way of measuring inbreeding. But the takeaway message here is that between 1990 and 1999 in Holsteins, we had, regardless of if you were looking at per year or per generation, we had a relatively high rate of inbreeding. Okay, then the period came between 2000 and 2009. Remember I had mentioned that R value that came out and was so cool and everybody used it apparently. Then 2000, it sort of reduced then. But then 2010 to 2018, genomics happened. And we got on this really fast train, this like high-speed train. And look what happened to all the different types of inbreeding. Now, blue here, this blue one, the lowest one that looks best, is what Lactanet publishes. And I love Lactanet, don't get me wrong. But you guys are kidding yourselves if you're only using pedigree inbreeding. Okay, that's a harsh truth. Pedigree inbreeding will give you an idea, and it's comparable, but it's not going to give you the real level of inbreeding, the real level of homozygosity in your animals. Okay, all these other colored values here, or most of the other ones, we'll put it this way: the low ones, red and blue, those are based on pedigree information, and the higher ones, that's based on DNA. Okay, so this is Holstein. You're like, oh, Jersey's gonna be way better, right? Yeah. Okay, so Jersey was way better. And is way better because it's not quite as fast. It's not quite as nasty. And you guys even achieved between 2000 and 2009, even got below 0. So that means that you were actually increasing diversity for a little while, just a little bit. But now, your pedigree inbreeding and your genomic inbreeding looks pretty much the same. And that has to do with this data set because it's smaller than the Holstein data set. But at the same time, I'm going to guess that your population structure is a little bit different from the Holsteins, right? You guys don't breed cows like a Holstein breeder does, I think, right?
Do we also register, as a percent of time, register our cattle more? Like there's more grades?
It could also be. And there's going to be a whole new, I heard you guys have already been discussing about breed purity and stuff like that. We don't want to talk about that today, but that is also associated here with this discussion, right? You have to think about all of these different factors, but this is interesting to see, for sure. So I'm a terrible person because I realize that this is actually, these are Holstein numbers, but the effective population size, if you look at pedigree, is 66 for Holsteins. I think it's 89 for Jerseys. That means 89 animals represent all of the diversity in the population, theoretically, right? 89 animals doesn't sound like a lot, but 66 is even less. You can take consolation in the fact that the Holsteins are worse than you guys are, but for a bigger population. And with regards to what's like really dangerous, FAO says 50 animals is like the below that, you're almost getting to you can start thinking about the word endangered, sort of down there. But that's not the case because we have a different situation in livestock, right? We're using AI. We've got cryopreserved semen and embryos. Yes?
Just for comparison's sake, what would the effective pedigree size be for chicken?
for a chicken line, right? Well, actually, if I go back here, this data, I just used this graph here to show the picture, but actually this is turkey data. And you can see the inbreeding here is like at 0.3. The average inbreeding coefficient is 0.3. The average inbreeding coefficient for dairy is like, if you use Pedigree, it'll be, you know, pretty low. But if you use Genomics, it'll be a little bit higher. The difference is that I'm going to guess it's around 30, 40, depending on the line. Long story short. Technically, yes. Right? Maybe we need, and this is the thing, right? We're in a totally new era. We have to be a little bit aware of that as well. That we're living in a time now where things are very different than they were at the time when Sewell Wright was proposing the idea of inbreeding. Right? Very different. And just alone in the last 10 years, we've had this massive change in how we breed animals that we just have to be aware of. Okay? So, conclusions here, rate of inbreeding are increasing faster since genomics. Implementation of genomic selection is decreasing effective population size. Number 2. Number 3, maintenance of genetic diversity could support long-term genetic progress. Could. Maybe. Right? Number 2, study 2. We want to see what does inbreeding actually do to production and fertility traits, and what is the effect of recent and ancient inbreeding on production and fertility traits. This was published in BMC Genomics 2 years ago now. And unfortunately, we didn't have enough data for jerseys to do this with Jersey data, but we did it on Holsteins. Okay? So this is a really ugly table, but ultimately, we looked at pedigree, ROH, runs of homozygosity, and another type of genomic inbreeding. And the only thing that I want you to take from this slide is that across the board for all of these traits, milk yield, fat yield, protein, age of first service, number of services, non-return rate, first service to calving, number of services, non-return rate for cows, across the board, there were differences in the group of animals that were really inbred and the group of animals that were not inbred. There were differences in like basically all the traits that we looked at. And the more inbred the animals were, what's your guess? Negative effects, right? But still not negative enough to say that the genetic gain that you're making is not worth it, okay? Just keep that in mind. There is a question. Some people say, oh, well, we don't have to worry about inbreeding, we just chop the pedigree off. Anybody hear that? Brilliant, right? 2 generations, that's fine, right? Not fine. Number 1, not fine, right? This here graph shows recent inbreeding. Recent means like in the past couple generations, right? You can see here these guys share a common ancestors here. This red piece of DNA came down through 2 branches of this pedigree and ended up in this animal. Recent, pretty recently. Now let's imagine like 4 more generations pass, and then somehow this little piece of red DNA happens to land again in the same, one single individual. Okay, that's old inbreeding, let's say. Now, what do you think is worse? Recent inbreeding or old inbreeding? Who says recent? One person. Who says old inbreeding? Who doesn't care right now and just is trying to digest their food? That was really good, by the way. So some people say recent inbreeding is bad, some people say old inbreeding is bad. And it's a hard question, right? But we looked at it, and we found out that recent inbreeding actually has a more negative effect than old inbreeding. Old inbreeding even has a positive effect on traits. And you think about that for a second, and you wonder why that is, and then you think, it's clear. Selection is old inbreeding, right? The whole point of genetic selection is inbreeding. You're trying to pick the best, and you're reducing the amount of animals that you're using. The problem with new inbreeding is that the genome doesn't have time to deal with all the crap, basically. Please don't quote me on that. But it's kind of the way that it works. Yes?
This may be convoluted, but is the new inbreeding, is that looked at on animals that had no inbreeding already?
That's the question. That's a good question. And you try and base it on, if you're going like 30 generations back, you're trying to find animals that aren't that related, and then you've created a loop in the pedigree that's very recent. So you're trying to pick out the examples that that are really truly recent compared to old. The reason that this happens is because if there's a nasty mutation or something going on, if it's new, it's going to have an effect because it hasn't had time to die out. Or the animals that have that mutation don't have time to die out. Now, the whole thing that I told you about genomic selection and that we're going so fast, that's having an effect on how quickly these nasty alleles get driven out of the population, something called purging. So we've got to be a little careful here, right? You can see here for, what is this? Effective 1% increase in new and ancient inbreeding. This is milk yield. So new inbreeding, milk yield has a detrimental effect here. It goes into the negative. But old inbreeding or ancient inbreeding has a positive effect. Yep?
So doesn't our system of indices drive new inbreeding? Like numbers beget numbers, right?
Beverly, that is a fantastic point. Yes. Who looks at top lists?
Narrowing our genetic base on the outside.
Absolutely. Who's looking at top lists? Do you always take the top? Not always. That's good. Who always takes the top? Why would you take like vanilla when you can have chocolate, right? It's a good question. I mean, not the vanilla chocolate thing, but you know, if you've got this fantastic list with top bulls, why not just take the top because they're the best?
Effectively, we're all taking the top. I mean, you're not taking the top of the top, but you're still taking from, I mean, it's all of us. We're taking, because AI companies are already only taking the top 1%, so we're all taking the top.
Exactly. But we have to know that, right? We have to know that a little bit. And this graph here is for milk yield, age at first service in heifers. This one even gets stars about how different they are, right? So this is new inbreeding and ancient inbreeding, or old inbreeding. So there's a difference between new and old inbreeding. Conclusions here. Negative effective inbreeding was observed on production and fertility traits. Recent inbreeding had more detrimental effect in comparison to ancient inbreeding, and no substantial evidence of purging due to selection. So selection pressures don't have time in today's world to sort of knock out what's happening, okay? Third study, we wanted to find specific genetic regions where homozygosity has negative effects on production and fertility traits. So that last study was pretty cool, but we said, all right, we can do better. We can look at the DNA and find out where exactly in the DNA are there spots that are homozygous and have crappy effects on phenotypes. We want to see if we can actually localize those spots with genomic information. And this was published in Genetic Selection Evolution, I think last year. So again, and this is just using Holstein data again, because we didn't have enough to do the Jersey stuff. So we found a bunch of runs of homozygosity with extreme effects, with extremely unfavorable effects on milk yield, fat yield, protein yield, number of services, non-return rate, first service to calving. Those effects were found on chromosome 8, 14, 6, 1, and these were the different effects, and this was the significance level of those effects. Main story here is that there were specific spots in the DNA that we could identify that had a negative effect. And interestingly enough, there were regions that had a harmful effect on multiple traits at once. So not just individual traits, but like a bunch of traits together. So milk, fat, protein, and number of services. There was a spot on chromosome 19 at 23 million base pairs to 28 million base pairs. So that's actually the physical distance in the DNA. That chunk of DNA had a nasty effect on all 4 of these traits at once. We're getting better at this. We just have to keep looking at more data and have more better phenotypes. But in any case, what we found is that there are unique runs of homozygosity regions with unfavorable effects within and across traits. Candidate genes with negative association were detected, and we have to start thinking about this a little bit more than we have probably in the past. Now, I'm not like trying to freak all you guys out, but I'm just saying, like, maybe we should think about this a little bit, right? So what do we do now? That's the real question, actually, right? And to think about this question, we have to think about all of the different perspectives that are flying around in our wonderful world. And the first perspective is from the industry, right? If I ask you, what do we do about inbreeding? What would you say? You, yourself, and I. No, you.
From an AI point of view, we service a lot of people with a lot of different diverse ideas as to what the ideal animal is. I can go around the room here and I can point to people that I know have a completely different idea of what a good bull is versus this person. As you drive down that road of what your ideal bull is, then you're going to do exactly what we talked about before. You're going to write a tree because in order to get, so if you take a high-tight bull, in order to get high-tight, you limit your bulls and your selection to your high-tight. If you're going for high production, like some of the genetic bulls, you're going to narrow your production because that's a different segment. So what you need to do is open it up to both, have a little bit more open-minded approach to what good bulls are, kind of interbreed them. And you also need to do more genomic testing to open up the base to get a bigger base. With opening it up to doing more genomics, you're going to find more ideal perspectives. Bull mothers, all that kind of thing. If you don't do more genomic testing, you're stuck with what we have.
You're going to do my presentation.
Exactly.
You're going to do my presentation.
So from an AI point,
Yeah.
that's where we need to go. The other thing from the AI side of things is because we got to service both.
Yeah.
We're kind of switching. We're neutral to what is ideal because we need to market both segments.
For sure.
And 27 years ago when I graduated college, we could sit in the rooms and we had the showdowns and everything with commercial guys. That 20 years from now, you're not going to be in business because both of them are wrong. Well, I can go back to those same barns and yeah, it's interesting that both won't be out.
It's the same now.
Yeah. So as long as you're efficient, we gotta take what's efficient. And everybody's ideal is different. Take that ideal cow and make more of it. So that's how we do it.
Okay. So generally, and maybe some of you guys are of that opinion, maybe some of you say, I do everything I can to avoid inbreeding at all costs. Is there anybody that resonates with that statement in this room? No. What about this one? I don't like to see an increase in inbreeding, but there's not much I can do about it. Yeah, okay, we've got 1, 2, 3, 4, slowly, there's more, 5, yeah, okay. And what about this? I actually don't give a damn. No, if it ain't broke, don't fix it. Who thinks that way? You can, it's okay, I won't tell anybody. You're only being recorded there, guys. But it's only the back of your head, maybe they won't be able to tell who you are, right?
There's probably a D in that monitor and assess as you go forward on a small scale within our herds, but yes, I'm aware, I monitor it. In some cases it's warranted if I can get what I want,
Yep.
but in other cases, if I don't need to, I don't need to. So I think there's probably a D there as well.
Perfect, diplomatic answer, yep.
We've kind of been pointed at 7% inbreeding as kind of a threshold or a percentage threshold for inbreeding. Does that hold weight?
What do you
as a
think
genetic
after this
lab?
after this presentation?
Still too
I don't know.
7% at your genetic gain maybe 10 years ago was maybe a number, but with how fast
Yeah,
we're
has
going
that
now,
changed?
maybe it needs to be far less than that before, because it increases so much more quickly, right?
And maybe it's not the percentage, but the rate of increase. That's probably more what we need to be aware of, right? How fast we're going. If it's going from 7 to 8 every 10 years, oh, maybe that's okay. But if it's going from 7 to 8 every 1 year,
Well, it's kind of like a snowball. Once it gets going, it's going to pick up.
we just have to watch it, right? And I love actually your D answer here, right? So this is really interesting for me to hear, because you guys are the ones doing it, right? You're the ones breeding your animals. But there are other groups who are interested in what you're doing, okay? Last year, I got in a hell of a lot of trouble because the Wall Street Journal called me and said they wanted to know about inbreeding. And at the time, I was doing this fantastic research project, and I had all these great students, and they were all into it, and they're like, oh yeah, this is so cool. Look at this, it's going up so much, and it changed from 2000 to 2010. So I was like, all right, I'll talk to them. And what they came out with was this article saying most dairy cows are kissing cousins and scientists are worried. Boy, oh boy, did I get in shit for that. Whew. Yeah, I don't have a real boss, but man, I've got a lot of funders who are kind of like, what the fuck are you doing? Like, really?
You get out in the country a little more.
But the statement also in a different article was the gene pool of livestock is about as deep as a pie plate. Okay, so what does that say to the 98% of the population that have nothing to do with agricultural production? What's that?
The media is stupid and they need to stop listening.
That'd be fantastic, right? But it's not the case. It's not the case at all, right? Because you've got people who are, and most of those 98%, maybe even they love milk, and maybe they think, you know what, our farmers are doing a fantastic job. And you talk to Mike Van Massow at the Department of Economics in the University of Guelph, he'll tell you the consumer has a fantastic view of agriculture, thankfully. The public is different than the consumer, right? And there are some who are not so rosy. But ultimately, if we take a long, hard look at what it is that we're doing and what's driving our decisions, we want money, just like everybody else. We're driven by gain. And that's okay, because we have to be, but we have to do that responsibly, right? I don't think that this is, well, I'm not even going to say what anyway. So industry perspective, right? Public perspective. And then there's the academic perspective. And this is the big, long, complicated, convoluted statement saying the benefits of intense directional selection currently outweigh the detrimental effects of inbreeding, but there could be a threshold at which lack of genetic diversity causes serious problems. And then in a typical academic fashion, you end saying, we don't know when and if that could happen.
That's what you told the Wall Street Journal.
That's actually what I told the Wall Street Journal. That's actually what I told the Wall Street Journal, right? And yeah, and it's what I'm telling you now because I stick to that. That's true for me right now. We don't know, but we have to watch and we have to be a little bit aware of this. Yes?
I saw Mike Van Massow speak a couple weeks ago.
Yep.
And he said he found out 20 years ago there is no the consumer.
Yeah.
Have you heard him say that?
I've heard him say that.
There's so many different, there's no average of consumer. They have a lot of different views.
Absolutely. And it's not just the consumer, it's also the public, because some people don't buy products and they're not a consumer of ours. They don't drink milk, nor do they ever want to. They only eat things that fall from trees. I'm just being really, really nasty there. But like, really, these are people who don't have anything in common with many of us, who have different views, different morals, different ethics, different guidelines, different thoughts on everything. And that's okay, because we're all human and we can do that. But everybody's going to have an opinion, right? You can't shoot everybody, right?
But their gene pool's pretty tight.
Might be.
They're all under the same tree. Genomics has helped identify those things.
Absolutely.
And those are good things.
Absolutely.
Going forward, because you're looking for it and we're going to prove the base from what we're looking from, we're going to find more. That's not wrong.
Yep.
Because if they're managed properly through mating programs and things and such, you're not going to allow those potential matings to happen.
Exactly. The more we know, the more we can do about it.
HCE, when you're talking about that, if you go back and talk to Nathan Holsteins, he's identified which holes they work, so all of a sudden that makes sense. Those kids in that hole.
Yep.
Some guys couldn't keep them alive.
Yep.
Other guys didn't have problems. And some were mixed.
Yep.
But it all came back to the level of HCE carriers within that herd.
Exactly.
And then you put a prevalent side of it as a carrier, if you weren't measuring how to fix it.
Yep.
That's when it happens. Going forward, as we identify more, and we manage through that, it's also going to decrease that. You'll have more carriers in the population, but if you manage them, you're going to eventually be
Purging, right? Our selection, we have to, the train is going so fast right now, and we're all like, yes, genomics is so awesome, I can make so much money, it's fantastic. And all of a sudden it caught up to us, where we're like, oh, maybe there was some thought to put into proving bulls, right? Yep.
We're talking about purging, we're talking about managing, so what about like editing? Does that lead to the fix, is editing?
If you know exactly where you have to edit,
Yeah.
right? For those of you who don't know, genome editing is a technology that is up and coming, right? CRISPR-Cas9 technology. You can actually go into the DNA, into those snippets of DNA, and you can cut and paste, just like in a Word document. Take 3 letters out, put 2 different letters in, and boom, you're golden, right? That's possible. It sounds freaky, but it's absolutely possible. The question is not a technical one, it's a regulatory one still right now. Yes?
And doesn't it become a GMO?
Well, that's the question. And interestingly enough, genetic modification, depending on which definition you use, and this is where people get a little like foxy and they get all wiggly and stuff. They're like, oh, it depends what you use. But if you use the definition, the true definition, saying a genetically modified organism is one that would not occur in nature, then no. And the bugger of it all is you can't even tell. If you edit an animal, you can't even tell. So how on earth is anybody going to regulate that? I don't want to be responsible for that, right? Like, I know my wheelhouse, that's not in it, right? Yeah. So, what's the reality after all of these different perspectives, right? We've got all kinds of already industry perspectives, we've got public perspectives, we've got academic perspectives, we've got student perspectives, we've got the 40 million consumers, right, who all have different perspectives. We've got the vegans and the animal rights. We've got like lots and lots of perspectives. The reality is that inbreeding is unavoidable in intense directional selection programs. What does that mean? When we're going for a goal, intense directional selection. We have a direction because breeding organizations set the breeding objective. That's what breeding organizations do. So you have to have a direction that you're moving in, and we're moving intensely in that direction, and our genomic train is going there very fast. Okay? That's reality number 1. And reality number 2 is that the diplomatic answer that came from Larry earlier, at best we should manage and monitor the loss of genetic diversity, because it's a loss and it's happening, and we have to own it because we're responsible for it. Right? So, there's a breeder's dilemma here. I don't know if you guys have ever heard of the breeder's dilemma. It makes me not sleep at night. It's really terrible because you've got genetic gain on the one side, and you've got genetic diversity on the other side, and you've got these 4 sort of quadrants, right? The top one here, you've got lots of genetic gain and very little genetic diversity. That's risky, in my opinion, right? Then you've got low genetic gain and low genetic diversity. That's just stupid. Excuse my French. Sorry. but right? That's just really stupid. Then you're just worried about genetic diversity, but not getting any genetic gain. Some people might say that's stupid too, but actually that's stable, right? You're not going to make fantastic leaps and bounds, but you're going to be just chugging along all the time. Ideal is here, where you've got lots of genetic gain and you can keep as much genetic diversity as possible. That's the golden ticket right there. And there are tools and strategies for doing that. These tools and strategies are available. If you Google software for preventing inbreeding in livestock, this is what researchers do, by the way. That's how we do our research. I'm just kidding. We actually do real research, but I did this, like, I don't know, last night after I had that fantastic experience of doing an auction with you guys. I was poor, but I was still Googling. There are about 127,000 results there. So it's not that people haven't thought about this before. This is not a new question, right? And there is lots of groups, lots of my colleagues work on something called optimal contribution selection, where you're actually trying to find that ideal spot between genetic diversity and genetic gain. There's lots of programs. AlphaMate is one of them that came out of the Roslin Institute. There's the Australians have done lots. Everybody has these programs that you can use. They're everywhere, right? What's the reality? Anybody know? Well, it's rarely used in practice, right? And the top lists dominate conversations, decisions, and semen tanks. Top lists, and I don't mean like the top 1,000, I mean like the top 100, right? Outcrosses are really interesting, but nobody actually buys them, making it really difficult for an AI company to invest money in buying outcrosses, because if you guys don't buy it, because you're like, well, why would I do that, really? And people, and I heard last night at the banquet, lots of people are doing kind of experiments, right? They're saying, oh yeah, I'm going to cross with this or with this or with that. Yeah, you can do experiments, for sure. But that's not the population of Jerseys in Canada. That's like one herd and like maybe 10 cows or something like that. That's not going to change the population structure that we have in Canada, of our Jersey population, or in North America. Oh, that was scary. Oh. So ultimately, there's two critical decision points here. And that affects the diversity of dairy. Not only Jerseys, but like of dairy. And of livestock, really. And this problem is a chicken and egg problem. Because who's responsible for maintaining genetic diversity in the cattle population? Ultimately. And now you can hear a pin drop. This is so fantastic. I'm so excited. Right? Are you all excited? Who's responsible? Well, what bulls do AI companies purchase? And what semen do producers purchase? It all comes down to this. Because as we all know, like 90% of the genetic gain is made through the male side, but an AI company won't buy a bull if you won't buy it from him or her or whoever. So it's back and forth. It's back and forth. And there is no way in hell that an AI company is going to buy all outcross sires and go to market and say, look at what we're doing. This is so fantastic. Because nobody's going to buy it. And everybody else is going to be like, haha. Right? You're done. One less to go, one less to compete with. Fantastic. Wonderful. And this is an interesting discussion because it's a back and forth, and it's a chicken and egg problem. And unless we deal with it, what looks to be a fantastic gain-making industry is in 20 years going to become obsolete because the public say, what on earth have you done to our animals? We got to figure this out somehow, right? I think. So what can we do? Like, what can we actually do about this? Well, I came up with 3 things, but I'm not by any means, well, I guess technically I'm supposed to be an expert, but you guys are the ones who are actually doing things, right? So the first thing I think is that we should include genomic information where possible for more accurate measures of inbreeding. Because it's more accurate. And it's more real. And we have it, and we can use it. And if you look at the inbreeding coefficients on Lactanet's webpage, they're based on pedigree information, because that's what we have for most of the animals. But there are some animals that have genomic information. In fact, there's like 300,000 Jerseys that I played with the data that I know that they have genomic information. So I think that that's more real and more accurate. I think that for you guys, it would be really useful if you had a tool that could provide you with a list of the bulls that are least related to your herd. And I was kind of scared to write the second part of that sentence on this slide because I'm not sure if I'm going to get lynched after this. But the second part of that sentence would be, comma, that is irrespective of your AI company and of your AI rep. And I'm so sorry. but I want you to think about that. Who makes the decisions on your farm? You do? Good for you. Do you really? And it was very recently, I had a conversation with a bunch of producers and I asked them, I said, who makes the decision? They said, well, I do. I say, okay, how do you make that decision? They say, well, I sit down with my AI rep. I say, oh yeah, who's your AI rep? And they say, well, it's John or James or Jim or Buddy or Matt or whatever. And yeah, I tell him what I want and he runs a program and I get my suggestions. And that's exactly how it should work and exactly how it does work, but maybe there's more to it. So my question to you is, would something like this, with the second part of that sentence on there, would that be interesting for you? Would you even look at it? Who says yes? That's a pretty certain yes there. That was a pretty high hand up. That's good. I like that because I'm blind, right?
And some, I mean, it's maybe not a high percentage, some do that now because we deal with 4 different AI companies because we're looking for genetics from around the world. I mean, we have access to it, you know, wherever we want to go and get it, whether it's Denmark or Australia or the US or wherever, we can do that in all the different AI companies. So some, and maybe it's not a high percentage, Christine, but there are a chunk of us that already do that and work with all the different. We don't have one company, we probably have our preferences, but it kind of depends on how the wind blows a lot of times. So that's already happening. What you're saying is we need to accelerate them and make them become more, everybody wants to sell, so they need to work together more to make that happen.
Does it really matter?
That's another question.
I mean, I would look at the tool, but the part about the AI, like, you're buying select sons and they're buying CMEX sons. Does it really matter?
Yeah.
Next question, right? Does it really matter?
So.
Does it? I don't know. My question is, what can we do that does matter a little bit at least?
From my experience, that first part, I don't have a problem with what you had said.
Yep.
The part that you got to add into that is everybody's got a different level of importance on different traits.
Exactly.
And like I said before, I can go, I'm not going to point fingers, but I can take you to guys' table that are sitting here now. They will not use a bull that is below 8 or 10 for confirmation.
Yep.
There's other guys that I can go to in this room that really don't care what he is for confirmation. They won't use a bull unless he's over 1,200 mil.
Yep.
Those are two completely different sets of genomic bulls to be looking at.
Yep.
So to really validate that second part, you've got to put in there bulls that are least related to their herd for the traits they want to use themselves.
Yes.
And that's where, because everybody's different,
Yep.
everybody's got an opinion on what they want within their herd, there is no cookie cutter answer for what it is that they want.
Exactly.
So if you can do that, I don't have a problem with that.
Yep.
Like, I think that's good.
Well, and yep. Oh, sorry. Yep.
Some of the things that you found about the negatives.
Yep.
Doesn't that make our genomics not accurate? Because if that's truly what happens when we do put those together, then it actually should be less than what that value is that's being reported. Wouldn't that actually help the industry to have those adjustments in there to make the numbers more accurate?
Well, it's a great point, but the economics are already in there. And that's what I mean when I say the monetary gain from intense directional selection outweighs the negative effects of inbreeding right now.
But for some of the indexes, don't we already have corrective factors for what we can estimate to be the inbreeding compression effect?
The US does.
Okay.
They do. We have for some, but it's based on pedigree inbreeding and it's very, very small. It's very, very, very small. And that was actually, of all of the like 40 resolutions that were given to Lactanet, one of them didn't go through. The one that didn't go through was that there should be an adjustment for inbreeding in the indexes. That didn't go through of 40. 39 that did go through, but that one didn't. Which is okay, right? But at the same time, there was another resolution that said, oh yeah, we have to do something about inbreeding. But I don't think that adjusting indexes does that, because I think they're two separate things, actually. I think. But it's absolutely true. There is no cookie cutter solution to this, but it's a chicken and egg problem, right? Because on the one hand, you guys have to buy bulls that people will buy, and you guys have to buy semen that's going to make you cows that make you money. That's it. And how to solve that is the next question. For me, as a professor at the University of Guelph, I'm going to continue doing research. I'm going to keep looking at these technologies to further understand the biological background of what homozygosity actually is, while trying to, maybe, I'm not sure if I should talk more to the Wall Street Journal or less, but we have to do something, right? We had a discussion about extension. It's difficult. It's difficult to do, cutting edge research and do extension and talk to the Wall Street Journal and. But we have to. It's difficult for you guys to milk your cows and plant your crops and take care of your kids and do all of those things that you do while still thinking about inbreeding. Maybe. It's difficult. It's not an easy subject, and it's a very complex matter. And with that, I'd like to, if any of you guys have ideas or other further discussion points, please contact me. We can talk about it. It was a pleasure to be here. Thank you so much to Lactanet and NSERC for the funding, and to my fantastic team, of course, of students. We don't just drink and eat, but we do a lot of that because it's fun. And we also like to do a lot of research. Thank you so much.
Any more questions from the floor?
Over the last 40 years, and this just isn't the dairy industry, but agriculture in general, our country being proactive has increased we've become more stringent at our borders to protect our domestic flocks, herds, etc. from disease. With that, how much of the outside genetic pool have we cut out of our domestic herds? Like, for instance, we can't do, embryos are okay, but there's some bulls in countries we can't access because of disease status, which I understand, I'm not against that.
Yeah.
But how much of our genetic gain have we limited because we're trying to protect greater good?
It's a great question. Genetic gain, I think we're doing pretty well. Diversity, I mean, so there's about, let's say in the Western world, there's about, and I'm going to use Holstein numbers, sorry, because I'm more familiar with those, but there's about 15 million animals in the herd. The effective population size of the Holstein breed, like all those 15 million animals, is still, if you look at DNA, below 50 animals. So less than 50 animals represent the entire diversity, and nobody cares if that bull came from Denmark, from the Netherlands, from if it's a Holstein purebred. That's the diversity that is represented in those 15 million Holstein animals. So yes and no, right? And then you can start to understand why the Wall Street Journal guy was talking about pie plates, right? Because it doesn't sound good. It really doesn't. But the Wall Street Journal guy doesn't understand that we have AI, that we have reproductive technologies, that we have cryopreservation banks, even at CGIL, the Center for Genetic Improvement of Livestock. Flavio Schenkel, the director of the Center for Genetic Improvement of Livestock, I don't know if any of you know him, but he was so happy on Friday. He was running through the department with a little wheelie cart with a semen tank behind him saying, "Look, I just got new semen." It's our responsibility. It was kind of weird. He was like, "I'm going to start my herd." But it's his responsibility. It's our responsibility at the university. We have a gene bank of all the young sires since like forever. And that's saved at the University of Guelph. There's also gene banks in a lot of other places too. So it's hard to say really, right? We're not like a normal ecological, we're not talking about like wildlife species where it really is endangered, like you're never gonna get that genetics back. We're aware of that. We might not be worried about inbreeding at all. We might not need to worry about inbreeding. But our public and our consumers hear that number 50, and we need to know about it, because when someone comes and talks to you about it, you need to know. Yes, we're breeding. Yes, we're reducing our environmental footprint by producing really efficiently and effectively. The number of cows in Canada has decreased from 4 million in 1960 to 1 million today, and we still produce the same amount of milk. Those are incredible strides in efficiency. The question is, do we, and I mean, you could be really provocative and say, do we even need diversity in dairy cattle populations? If you wanted to be very provocative, right? I don't know. These are questions that we can think about. But what I can tell you is that if we get to the point where the inbreeding load is so high that we lose fitness and fertility, and we lose the ability of our animals to adapt to perturbations, whether that be heat stress or environmental stress or anything like that, then maybe we went too far.